right, welcome back. In this lesson, we are going to be looking at Unit 6, Lesson 1, Exploration 3, and we're going to be talking about DNA replication. This is the last exploration of Lesson 1 and is going to finish up the structure and function and replication conversation about DNA. All right, now we've talked about replication before, meaning that we basically mentioned it when we talked about the cell cycle. If you think back to the cell cycle, you'll remember that during the synthesis phase of interphase, the S part of interphase, we said that the DNA is replicated, and we kind of just left it there. We didn't go into very much detail about how that actually happens because that's what we're going to look at right now. As you know, after that DNA is replicated, we now have two complete copies that can be given to the two new cells that are created at the end of the cell cycle. The cool thing about the way that DNA is replicated is that it provides a very accurate, very useful strategy to making sure that the DNA is copied quickly with little to no errors, which is huge because errors in replication can cause massive problems for the cell down the road. So let's look at how this actually takes place. This process is going to be accomplished using a series of enzymes. Now, I want to show you this clip, and I'd highly recommend that you go back and look at it. This clip provides a really great visual example of how DNA is copied, and it might not make a whole lot of sense right now, but after we've talked about this and after you've read through this, this clip is going to be something that you want to go back to with an understanding of how these two strands are replicated. So I'm going to play this for you. This is the strand as it exists when it's put together. When replication begins, the first thing that's going to happen is that DNA molecule is going to be unzipped. And that's what you see right here with this red protein enzyme. It's unzipping it into two parts. Now, we'll talk about this in a little bit, but I want you to see how these two parts are synthesized a little bit differently. One of them is pretty much just synthesized in one giant piece all at once. As it's opened up, new pieces are added to it, and it becomes a complete strand on the other end. The other side is happening a little bit differently. It's kind of being built in chunks. You kind of see a chunk right there, another chunk right there. It's being built differently because of the way that DNA molecules fit together. Their shape, as we talked about in our last lesson, is anti-parallel. They go in opposite directions, kind of like two sides of a two-lane highway. And because of that, the new strands have to be built differently where they are opened up. So let's look at how this happens. There are going to be 4 enzymes that I want you to be very familiar with. Again, by the end of this lesson, you're going to have 4 enzymes that I need you to know what they are and what they do. So we talked about the enzyme that's responsible for unzipping that DNA molecule initially. That enzyme is something called helicase. Okay? Helicase is that enzyme that unzips the DNA molecule and starts the process. As you can see here, there's a helicase enzyme, and it's moving kind of this direction, opening that DNA molecule as it goes. There's another helicase over here. It's not labeled, but it's the same pink structure, and it's moving to the right, opening the DNA molecule as it goes. These balls right here are stabilizing proteins. They keep the DNA open long enough for the new strand to be created. Otherwise, as soon as helicase opens this up, the DNA molecules will just close right back together because of those hydrogen bonds that are attracting to each other. So this is how we open up the molecule. The enzyme responsible for that is called helicase. It breaks those hydrogen bonds, exposes each side of the strand so that a new strand can be added onto it. Now, the process of creating a new strand of DNA is possible because of the complementary nature of the nitrogen bases. If you remember, DNA is made up of 4 nitrogen bases, A, T, C, and G. And those nitrogen bases always exist in complementary pairs, A with T, C with G. So when this unzipping process starts, what we see is an exposed side of a strand. Now, that strand is going to be fitted with the complementary base pair. Whenever there's an A on the template strand, a T will be added onto the new strand and vice versa. And that's how these new strands are built. Now, this picture provides a really good shot of how this happens on both sides. If you want to focus on one side, you just click to a different picture. Okay, remember how I said that the two strands are synthesized differently? This top strand shows you the strand that is just built in one piece. There's one starting point, and then the nitrogen bases just get added as it goes because that's the direction of the new molecule. But on the other side, this direction gets built in pieces. As it opens up over here, this is where the first opening happened. As a piece gets exposed, once there's a little bit of an exposure, that gets closed by new DNA. Then it works back to this spot and closes, works back to this spot and closes. And this takes a little bit longer, and there's a little bit more involved with it. But this is a great picture to study to be able to talk about this process. And again, your textbook is full of all kinds of great graphic organizers. This one is particularly great because it talks about the 4 different types of enzymes that are involved in this process. Now I want to go back up here because there's a couple things that we need to talk about. First of all, an enzyme called DNA polymerase. It showed up earlier in the lesson, maybe right up here, yeah. DNA polymerase is the name of the enzyme that is responsible for adding new nucleotides to the strand. Remember, a DNA molecule is a group of nucleotides put together, and DNA polymerase is the enzyme that's responsible for building that new DNA. It's responsible for building that new strand. So in this process, we have a couple key players. So I want to go back to this diagram to talk to you about what they are. Okay, first of all, helicase. We talked about how helicase is responsible for unzipping that DNA molecule. Okay, now before we also talked about RNA polymerase right here. So we know those two. All right, so then there's 2 other ones that we haven't yet talked about. So let's go back to this picture. All right. In this picture, I want you to see this right here. It says RNA primer. The RNA primer is an enzyme that puts down a small piece of RNA that basically signals the starting point for new DNA to be built. Without the primer, DNA polymerase doesn't know where to begin building the new strand. So RNA primer adds the starting line for this whole process. So we can go back here. This primase inserts the RNA primers. And then the last one is an enzyme called ligase. And ligase exists on this second strand right here, the strand that is not built in one piece. The key idea about ligase is that ligase can connect these segments together. Because this strand is built in segments, it has to be connected after they are laid down. And so that's what DNA ligase's job is. It kicks off the RNA primer and it adds DNA to that spot to glue it together and make it into one solid piece. So again, back here, ligase's job is to connect those fragments. Now, I told you there's 2 strands. There is a name for these 2 strands. The top strand right here that's built in one piece is something called the leading strand. That would be this picture right here. The bottom strand that is not built continuously, that is built in chunks, is called the lagging strand because it kind of lags behind and takes a little longer. And it is on this lagging strand that DNA ligase has to connect those pieces together. All right, so again, I would highly recommend that you go through this and fill in this box right here because a basic understanding of these 4 players is going to be really important to your understanding of how this process happens. Now, we've talked about this and we've showed you the 3-dimensional computer simulation. That was this video up here. And we've also shown you a couple of 2-dimensional pictures. Now, the 3-dimensional video is awesome if you understand what's going on. So I'd highly recommend, again, go back and watch this. Use the pictures below to kind of make sense of what's going on. Try and figure out where all of those major players are located. That way you can describe this process. All right. When this process is finished, what we're going to be left with is 2 identical strands of DNA, which is, again, is important because that's what preserves the instructions for how to make all the proteins for that organism. Now, we call this process semi-conservative because each new strand is built from one existing strand of DNA. There's an original strand that we use to add the complementary strand to it. And because we're doing that on both sides, we get 2 identical molecules of DNA. Now, let's finish up with this. This process happens pretty fast. There's about 50 nucleotides that are added every second when a DNA molecule replicates. Problem is, there's a lot of nucleotides. And if you just opened the DNA at one spot, it would take way too long to build a new strand. So what we do is we open the molecule in several spots and start building in opposite directions. You kind of see that in this picture right here. In this picture, there's 3 different origins of replication that are going to begin spreading out in both directions, ultimately creating more opportunities for this molecule to be replicating all at the same time. And because of that, like it says in your textbook, this whole process only takes a few hours rather than days if it was only opening on one spot. Now again, this process is super cool because it happens very quickly and with an incredibly small number of errors. DNA polymerase has the ability to spell check itself. It's kind of like when you send a text message or when you send an email. There's an autocorrect feature along with most of these things that are going to catch a lot of the errors. Now, it doesn't catch everything, but it does catch a lot of things. There's an error rate of about 1 in every 1 billion nucleotides that are added, which is substantially better than any of the autocorrect mistakes that you're probably familiar with in a text message. So this process is really cool, very reliable, with very few errors. There's another video here. I'm not going to show that to you right now, but I would highly recommend you watch that on your own. Shows you this process of spell checking the DNA. All right, that's it for this one. Please go back, watch each of these videos, go back, look at the 4 different parts, make sure you label them, and ultimately make sure that you can tell the story of how this DNA molecule is able to copy itself from start to finish. All right.